Well, good evening, everybody. Welcome to the Immunisation Coalition's third webinar series on COVID-19. This evening, we're going to talk about vaccine development for COVID-19, and we're providing this to healthcare professionals and we'll have lots of opportunity to have questions raised and answered by the experts who are presenting tonight. The program is hosted by PraxHub and presented by the Immunisation Coalition and intended for use by health professionals. Before starting and introducing myself and then subsequently the panellist, I would like to acknowledge the traditional custodians of the various lands on which we work today and the Aboriginal and Torres Strait Islander people participating in this webinar. I pay my respects to elders past, present and emerging and celebrate the diversity of Aboriginal peoples and their ongoing cultures and connections to the lands and waters of Australia. I will be your chair tonight. My name is John Litt. I have been a member of ATAGI. I am currently a member of the Scientific Advisory Committee of the Immunisation Coalition. My career was in academic general practice and in public health, and I've had a long interest in vaccination, particularly in adults and older people, and prevention of vaccine preventable diseases. So before we get on to our first speaker, I'd like to introduce Gary. Gary is a director of the Immunisation Coalition and had a long and illustrious career in the Therapeutic Goods Association, where he was the director of immunobiology. So he was the person who had the technical expertise that when new vaccines came to the TGA to be evaluated for efficacy and safety, that he and his team were the group that did that work for the TGA. He actually had a number of roles with WHO, including the Health Systems Innovation Group, the Global Action Plan for Influenza Vaccines, the Essential Medicines Program, and led the Pandemic Influenza Preparedness Framework Review Group. He's currently an adjunct professor at the University of Sydney in the Faculty of Medicine. As I mentioned earlier, he's actually a director of the Immunisation Coalition. Without any further ado, I'd like to pass the baton across to Gary, and Gary's going to lead the charge on what we're going to talk about around COVID vaccines tonight. Thanks, Gary.
Well, thank you, John. Thanks very much for that introduction. And I need to begin just by declaring a few conflicts of interest. As John mentioned, I'm a board member, director for the Immunisation Coalition. And I'm also advising a number of groups, including a vaccine project at the Doherty Institute, BioIntellect, and also the World Health Organization. So I'd like to begin tonight by presenting a number of snapshots on COVID. So looking a little at the epidemiology, then the approaches to COVID vaccines, and I have a bit of a regulatory eye on this as well. A brief overview of the regulatory system. I'm going to concentrate on live and killed vaccines and nucleic acid vaccines, the DNA and mRNA. Paul will look at viral vectored vaccines and protein vaccines. I'll have a few things to say about adjuvants, but Paul will say more, and then a few conclusions and a summary. But to begin with, I'd like to bring you all in via a poll and see what your views are of this. Vaccines to COVID-19 are essential because of the need to protect high-risk groups, of the need to generate herd immunity, of the need in developing countries, treatments are unavailable, or correction, vaccines to COVID-19 are not essential. And as usual with these things, you do get varying opinions. So the two most favoured ones there are the top two: need to protect high-risk groups and the need to generate herd immunity. Well, let's have a look at a global snapshot and we'll come back to that. If we take a global snapshot of COVID as of yesterday from the World Health Organization, we have 42 million or so confirmed, laboratory confirmed cases and nearly 1.2 million deaths around the world. And it's absolutely sobering to realize that here on August the 5th, we have some 200,000 cases. By September, there were 400,000. And now towards the end of October, we have 42 million. This implies there's also a very large fraction of asymptomatic and mild cases that go undetected in the community. And some people estimate that fraction as being as high as 100 to 200 million people. The Centers for Disease Control estimates, and this is the good news, I guess, in a way, that people under 65, there's a 99% recovery, those over 65, a 96% recovery. And in Australia, we've seen, as of yesterday, some 27,500 cases with 25,000 recoveries and 905 deaths. We've also seen these two waves in Australia. In fact, you've seen this all over the world, particularly in the Americas and Europe, where a significant second, if not third wave is going through the community in those parts of the world, and now India as well with a significant second wave. An interesting way to look at things is via a heat map. And this heat map shows that the virus has spread all over the world, particularly in the Americas, EU, the UK, and India. It also shows cases and deaths per capita, which is an indicator of spread and also how well we're doing with interventions. In the Americas, you can see the cases and death rates per capita are actually quite high. In places like Europe, it's a little bit less, but nevertheless, the UK and the EU, it's still very high indeed. Over in India, it's moderately high. In Australia and New Zealand, we've been incredibly successful, as has Japan and Korea and Thailand, in keeping the cases quite low. Sweden is an interesting one. I put that in because they had no interventions compared to the rest of Europe, and they did worse than any of the European countries. Well, this wasn't surprising, having minimal interventions. We do know now that early interventions certainly work. It's a good reminder that the virus itself doesn't actually spread as people think, but people spread the virus, and therefore restriction, hygiene, and education are very much key to halt the spread of infection, particularly in the first part of a pandemic, or if there's any other significant wave. So apart from epidemiological tracking, there's also genetic sequence data tracking, and this allows virologists to follow the evolutionary course and relationships of these viruses to each other, as well as any mutations. Now, this slide, we don't need to follow it particularly. It comes from GISA, just to say there are 150,000 genomes that divide into several clades. You can see a GR clade, an S, a V, an L, a GH, and a G. It's the G clades that are predominating around the world. But new viruses, like here in green, new viruses from this part of the world also appear, and these can be tracked, particularly for mutations, which is really, really important. We can also design good testing kits and good PCRs. So from that point of view, it's really important. And if there's a significant mutation affecting the spike protein or receptor binding domain, then a new strain might be needed to update the vaccine. So this kind of data is incredibly valuable. The next slide will show these various clades in different parts of the world. Hopefully the vaccine will protect against all the clades. Different clades you can see are dominating in different parts of the world. Now in Australia, we've seen a new mutation, and that's in the hashed part here. So this amino acid mutation at 477 could be significant because it's approaching the receptor binding domain and might affect S protein, and therefore might affect the vaccine, which most specific vaccines are directed to. So this kind of information is really important, and the work that GISA does really is quite extraordinary. So what is the traditional vaccine pathway? Normally, this is what happens between 2 to 5 years, 1 and 2 years, 4 to 8 years for clinical trials, 4 to 5 years for licensure. In parallel, manufacturers are making small amounts of the virus or of the vaccine, and then they scale that up to commercial scale and large scale manufacturing. All this normally would take years, at least 5 years, if not 10. In the laboratory, we get antigen identification, we get testing for safety, we go through phase 1 to 3 trials, which I'll talk about a bit more later. And then there's a very important interaction with regulatory agencies at various points to ensure that everything is on track and that safety and immunogenicity and efficacy are all heading in the right direction. So here's a schematic of how long it normally takes. And if we look at this, having made a vaccine, we then have to manufacture it. But manufacturing isn't as simple as just turning on a tap. Apart from R&D and regulatory approval, we go through these more complicated pathways of raw material and bulk antigen manufacturing, and then formulation and filling and freeze drying, if that's part of it, packaging, lot release, and all the way, and then distribution, and all the way through in these little certificates, there are various controls, either in-house controls, which are really important for quality and safety, or regulatory controls. So it is normally a long process. And if we take some real life examples, we can, starting at the bottom, look at polio. Well, it took some 60 years to develop a vaccine, but in fairness, we also had to invent cell cultures. Ebola took 15 years. SARS-CoV, we've won. The original SARS, we haven't made a vaccine at all the attempts have failed since 2003. MERS failed as well in the last 6 years, and we're expected to make a vaccine in 12 to 18 months. Well, most people certainly question that, whether that was possible, but nevertheless, through various programs and a lot of money, there's certainly been an enormous effort by manufacturer and governments and various agencies to ensure that a SARS vaccine might be produced. The timeline also has to be flexible. It is possible to fast track vaccines from a regulatory point of view, and many of them do have such pathways for emergencies. This is from the TGA website. Even in their pathway, which I don't particularly want to go into, I just want to point out that it still takes at least 20 working days and up to 6 months is what the TGA require. And we'll certainly see for the FDA something similar. They need time, obviously, to assess data, which is extremely important. That part can't be bypassed. So here is the usual track. And Paul will expand a bit more on the various phases, but for preclinical, we normally see safety and immunogenicity, then dose determination and immunogenicity, and then safety again, and then efficacy and safety for phase 3, and then a continuous monitoring program for adverse events and effectiveness, which is phase 4, and right through the whole process, there's regulatory audit. So what are some of the problems that might occur? Firstly, we are combining phases, so speed could be an issue. Secondly, we have novel platforms and novel vaccine candidates, which are going to be given to healthy individuals. Some of these platforms have never been registered before, and none of these vaccines have ever been made before. They've never been licensed. There's not much manufacturing experience, and it requires, in some cases, new manufacturing facilities and specific lot release. Then there's a whole issue of production capacity, which is up to the manufacturers, obviously, and then formulations and rollout. And the usual issues will come back. We saw some of these in 2009, multi-dose vials. Do we use thimerosal? What about storage at 4 degrees? But mRNA vaccines will probably need to be frozen. How do you distribute those? Needle and syringe supply. If that's part of the formulation, some are not using needles and syringes. So what about safety concerns? We have immediate safety concerns because we have this novel vaccine in a novel platform given to a healthy population. We need to know the rate of mild reaction. We need to ensure there are correct immune responses. If we get inappropriate T cell responses, that can cause vaccine-associated enhanced respiratory disease. We really need to look out for that. And antibody-dependent enhancement of disease as well. And then febrile convulsions, the rate of severe reactions, any long-term sequelae. And that's just the immediate safety concerns. And then there are long-term safety concerns, looking for rare adverse events. And these are normally neurological, but narcolepsy, chronic fatigue syndrome, Guillain-Barré, encephalitis, transverse myelitis, autoimmune diseases, blood clots, eye infections, severe rash. We've seen these before with other vaccines that have been experimental. What about the clinical response and other immunological issues? There are many questions to be answered. Reinfection and susceptibility, for example, is still being looked at. We don't know the infectious dose of the virus. Recrudescence of symptoms and long-term sequelae. We are beginning to understand the long haulers, as we call them. Then there are the correlates of protection. Role of IgA has still not been elucidated. There's plenty on IgM and IgG, but the role of secretory IgA in the lung is still a research area. Immunogenicity, efficacy, and effectiveness is to come, but everyone's expecting it actually to be pretty low to marginal. Then there are genetic factors. We've seen a few papers describing blood types, for example, being more resistant to the virus. Whether that's true or not remains to be seen. So here's just a little snapshot of the immune response. What I want to point out here is that so far we've seen for a mild case the innate immune response and the viral load occur before the onset of symptoms and then the classic adaptive immune response. But in severe cases, it seems to be the innate response seems to be delayed and the viral load's a lot higher, and then the adaptive immune response kicks in. That's meant that the immune response needs to be controlled. And in our last seminar on treatment, we tackled that through various steroid treatments and statins and other things that were spoken of then. But the fact is we've never made a COVID vaccine before. So what about success with animal vaccines is a good place to look. And if we look at animal vaccines, yes, they've been made to a number of species, but they have all these things in common, which is probably not good news. That coronaviruses undergo frequent genetic shifts and you need repeated vaccination or boosters. We expect the same to happen with the human situation. There's a short duration of protective immunity. We're seeing that already in clinical cases. And there's a lack of effectiveness of inactivated vaccines, which is not surprising. So what about human vaccines to coronavirus types? Well, we've never made one. And these are the 4 common human coronavirus types, but no vaccine has been successful. These normally cause, as you know, common cold symptoms. There are no vaccines, but we do know there's a short duration of protective immunity. So this is so far in many ways not such good news. So might be good to bring you all in again with another poll question. What do you think would make a successful vaccine? One that generates immunogenicity, one that's at least 30% effective with no serious adverse reactions, one that generates appropriate cell-mediated immunity, one that will reduce hospitalizations and severe disease, one that gives cross-protection to coronavirus types. And here are the answers. So one that will reduce hospitalizations was the most favored. I mean, there's no correct or incorrect answer here. So that's good. So let's see what will make a successful vaccine. What would regulators expect? I mean, this is just a little schema to show that it's important that both arms of the immune response are activated by the time any vaccine goes through with any system. We do need good cell-mediated immune responses, and we do need proper antibody responses. So both arms have to be activated. Appropriate CMI, appropriate antibody, acceptable adverse reactions. You would think at least 50% efficacy and capable of reducing severity and therefore hospitalizations. I think this is an important point. It needs to be accepted by the community and regulators. So education is important for the community and various groups, College of GPs, College of Infectious Diseases, nursing groups and so on. Everyone really, the whole community needs to accept the vaccine, and therefore that needs tremendous transparency. It also needs to be tested in a large number of people to give everybody confidence and show that there are few adverse reactions. So how many people should be in the trial? Well, normally for a phase 3, 3,000 people with a large control group is fairly typical. But for a novel vaccine, WHO suggests 30,000 people. And fortunately, most of these studies so far are involving 30 to 40,000 people. And WHO have a document, this one here, TRS 1004, to have a 95% chance of observing one instance of a serious event occurring on average once in 1,000, it's necessary to expose 3,000 subjects to the vaccine. So for 1 in 10,000 serious events, you would need to expose 30,000. So we're in the right ballpark, at least in terms of numbers, to observe serious adverse events with large-scale clinical trials. So where are we with vaccine development? There are an array of vaccines available where inactivated and weakened or attenuated vaccines are viral vectored ones, nucleic acid-based ones, and protein-based. So at the moment, there are over 200 candidates and at least 32 in clinical trials. Now, the ones we're going to look at will be the ones in phase 3 and those that have got limited approval. So these 17 are what Paul and I are going to talk about. So for virus vaccines, just this is really the traditional approach. These vaccines either use a weakened virus, an attenuated virus. These would be a live virus, and it would give a full immune response if successful. Inactivated vaccine is again whole virus grown in culture of some sort and then killed using a chemical or heat. And The difficulty here is you need large quantities of virus to start with, but this really is the traditional approach, and there are a number of groups that are doing this, but they all have potential issues. For example, for live viruses, the attenuated virus might mutate and reassort. It's still unproven and unlicensed, the production capacity unknown. For killed virus, often you require high titers of virus for an inactivated vaccine. Again, they're unproven, unlicensed, the production capacity is unknown. So, you may need an adjuvant, which I'll come back to. So who are the leading candidates? Well, it's easy for the live attenuated ones because we're all still in preclinical development, so we won't really see much until about 2022. But these are the 4 companies that are following the live virus vaccines. But there's 2 interesting things about them. One is that most of them are by nasal delivery, which would, in theory, give natural infection and full immune responses, and others are looking at oral delivery. And because there's high expression of ACE2 and serine protease for the virus, spike protein priming in absorptive enterocytes of the ileum and colon would occur, and you should get a decent immune response. And animal studies are going in the right direction there. And of course, that would be known needles. So what about killed vaccines? Well, there are 3 leading candidates. So I'll talk about Sinopharm briefly. They had 2 candidates in Beijing and Wuhan. They both did well in phase 1-2 trials. And one of those has gone into a phase 3 trials in the UAE, Peru, and Morocco. Efficacy and safety is still not published, but UAE gave emergency approval of the vaccine for healthcare workers, and the Chinese government gave approval as well, and it's actually been already injected into hundreds and thousands of people. So they expect to make 2 billion doses by 2021, but no published data on safety and efficacy, and not trusted by Western regulators and scientists, at least at this time, until we see some published data. Again, Sinovac from China has also made a vaccine. They have a phase 3 trial in Brazil, Indonesia, and Turkey. And again, Chinese government granted emergency use. It's being used widely in people, particularly frontline workers, and they expect to produce 40 million doses by March 2021. Again, no published safety data, not trusted by Western regulators and scientists. And then there's the Indian company Bharat Biotech. It's called Covaxin. They're coming into phase 3 clinical trials, very successful studies in monkeys and hamsters, but they won't be producing until 2021. And again, there's no published data at this time. So adjuvants also need to be considered from a regulatory point of view. What do? They optimize the immune response. They aim to enhance and accelerate and prolong a specific immune response, and they're very useful for vaccine dose sparing. I'm only going to just briefly mention the first 2 here. There is a wide landscape and possibilities of adjuvants, but mineral salts are normally used, such as aluminium or oil emulsions, and you might be familiar with MF59 or ASO2 or 3 as classic oil emulsions that are used. But they do need preclinical studies, just like vaccines. They need non-clinical immunogenicity data, and if the adjuvant itself is immunogenic, then there might be tests required for hypersensitivity and, of course, dose response as well. So where are we with nucleic acid-based vaccines? So we see with the nucleic acid-based vaccines that for a DNA vaccine, the way it works is that a piece of DNA, it could be a plasmid with the coronavirus spike gene or the gene of choice, is made to enter into the cell through a process called electroporation, which opens up the pores and allows it to go through into the nucleus. Then there's translation to messenger RNA, and then transcription and then translation into protein, and then an appropriate immune response. It's the same with RNA, except this is encased in a lipid protein. It's injected, it goes into the cell, and then viral proteins are produced, and then there's an antibody response. So RNA and DNA-based vaccines are safe in general, or thought to be, and it makes use of genetic material only and not the virus, but of course they are unproven. Some of the issues are they are unproven, none are licensed, you probably need 2 doses. Scale up is is unknown at this point, how many can be made, and then safety and long-term effects as well. And again, as I mentioned before, public acceptance is very important. So for some of the potential issues for these vaccines, there's some fear about potential integration into the human gene because they do go into the nucleus, but there's no great evidence for that, but it's something that still needs to be explored. Poor immunogenicity in human, and the electroporation device is generally required. And I noticed that Inovio has had their trial paused because the FDA is looking at their electroporation device. For RNA vaccines, traditionally fairly low immunogenicity. They're not very stable because they're RNA. Generally, they need to be frozen, and that raises different questions about delivery. So the leading candidates are these 5 for DNA vaccines: Inovio, Angus in Japan, Zydus in India, Genexin in Korea, and in Australia we have Technovalia, which is a trial going through Sydney University. But again, these are mainly in phase 1, 2, or early 3 trials. So we won't really know much about any of these until the middle of next year at the earliest. And while these trials are going on, it will take some time before we know what will happen. But just behind, the other arm of the nucleic acid vaccines are the messenger RNA vaccines. The timelines are somewhat better. So the leading candidates are Moderna, who clearly in phase 3 injected some 30,000 people. We know the vaccine protects monkeys from COVID. They are undergoing some patent disputes, which might be an issue. And they're manufacturing over 120 million doses by mid-2021. Then there's BioNTech as another leader. They're also in phase 2, 3 combined trials. They have 2 versions. They're both immunogenic in large numbers of volunteers, and they've expanded their trial to 43,000 patients. And they're now looking at children as young as 12. They expect to produce about 1.3 billion doses. Then there's CureVac, a group from Germany and the USA, again, a large numbers of people. They're using these Tesla technology, mRNA micro factories. This is a bench operated thing, the idea being, clever idea from Tesla, that you could simply send the equipment around and people could make their vaccines as if you're making coffee out of a machine. But I don't know how that would go from a regulatory point of view. And then manufacture over 100 million doses by the end of 2020. There are 2 other vaccines just behind them. Imperial College London and Articus in Singapore with Duke, and they have a self-amplifying RNA idea. So the idea is that the RNA amplifies in cells and then produces protein antigens, and in theory, a lot more protein antigen than a normal RNA vaccination, and then an immune response. But it will take time for some of these to come on board. The leaders are BioNTech and Moderna, and we should get data for some of these arms by the end of the year, but probably vaccine, I doubt, would be available until early to mid next year at best. So in summary, mRNA and killed vaccines are not likely to be available before 2021, and the live and DNA vaccines are unlikely to be available until 2022, assuming they're all approved and things go well. There are several killed vaccines from China that have been given approval with limited use, and these are being used in the field, but there's no significant published data. mRNA vaccines are well developed, they're well advanced, with millions of doses being produced already prior to registration. But there may be rollout difficulties if it needs to be stored at -80. And that's something to be worked out, are the logistics around that. The critical and safety efficacy data will begin to emerge after November. And I couldn't emphasize enough, I think with all these vaccines, transparency and community consultation is really, really important. But we are left with a number of clinical questions, and I'll finish with this. Will one vaccine be more effective and safer than another? We don't know. Will the preferred vaccine protect against all the clades that are around, including ones that are about to mutate? Again, we don't know. Will the vaccine protect those at greatest risk? Children, those with chronic infection, pregnant women, minority and ethnic communities. The difficulty is that most of these trials don't have these arms. These arms are underrepresented in trials. Will the virus mutate and force new vaccines to be made? Almost certainly, but we just don't know when. Will vaccines prevent transmission? We hope so, but that's unlikely. And we still don't know the correlates of protection. It might prevent severe disease, but it doesn't look like it will necessarily prevent transmission of the virus. Will vaccines prevent severe disease and hospitalizations? Well, that's the hope. And will vaccines give long-lasting immunity? We already know that probably not. There will booster doses will be needed, probably annual vaccinations, a bit like influenza. Will the community accept the vaccines that might not be optimal? And that's the bit where where transparency to the community is important to give confidence. And finally, what about priority groups for rollout? And that becomes a matter of government policy, and I won't particularly comment, but normally I think most papers are saying that it should obviously be healthcare workers would be one of the groups up there. Then obviously people in institutional settings and those at high risk, you would think, would be two groups that would receive priority. So I'd like to acknowledge Jenny Hertz from BioIntellect, the GIZAID group, and John McEwen and WHO colleagues at the World Health Organization who provided information and some slides. And I'd like to hand back to John now to introduce Paul.
The first question comes about vaccine-associated respiratory disease. We saw that with the initial SARS-CoV vaccine when it came out with SARS in 2003. The question particularly is being asked, how long after getting the vaccine would you see that? I mean, presumably people are going to be watching for that, but is that a sleeper condition, or are we going to get the answer to that from some of these studies?
No, I think the studies are large enough to answer those sorts of questions. When you have 30 to 40,000 people, as long as you have several arms, remember if the arms are very, say you have 18 to 55-year-olds and that takes up 90%, then you've only got very small arms in pediatric and people over 65 or risk groups, which is necessary, but in a way a bit of a mistake. We need to also have decent arms, decent numbers in other groups, but most of the vaccines are actually targeting healthy people 18 to 55 or 60. So more trials will have to be done in other groups, but I think this thing is large enough, studies are large enough to be able to pick those severe adverse reactions. Good.
There is another question about the Oxford AstraZeneca vaccine, but I'm not sure what the question is. So maybe the person can to respond to that. Can I just pick you up with the groups? Being in one of those older age groups with higher risk, and I actually did a survey of some of my GP colleagues, and a number of them in a similar age group, they're particularly concerned for the older people because you've already alluded to the fact that they're the group that have the highest mortality and the most likely to end up in hospital. And you've just commented that this is a group that are not having the vaccines, they're not the main target for the vaccines. So, I mean, most people are familiar with immunosenescence. Can you just expand on that? Because I think quite a few of my GP colleagues asked about that group because they're getting asked by their patients, will this vaccine protect me? I'm 70, I've got blood pressure, I've got type 2 diabetes. Is this going to work?
Well, see, normally this is a problem of combining phases and regulators will have to think about it. But when you start combining phases, then you tend to leave certain groups out. Now, normally you would do a study in healthy adults, then you would step up, so to speak, to an older age group, then you would step down to a pediatric group and maybe step down further to under 5-year-olds. Then you would take time to look at pregnant women. You would take time to look at people with immunological conditions of some sort. You would take time to look at ethnic groups. And that's one of the reasons it's often an 8 to 10 to 12-year project to make a vaccine that you're very confident about with all the various groups that you want to protect. Now, this has all been sped up tremendously, but we will get good data from my reading, at least on the vaccines I've talked about for the 18 to 55, and we'll get some data for the other groups, but hopefully it'll give confidence and there won't be any problems, but then it will be up to regulators to decide, yes, there's a vaccine and it can only be registered for this age group or that age group or in these circumstances. Now, all the studies are very different. They're not all the same. And so some have got an arm or two with reasonable numbers, but it won't be 30,000. There's nobody doing a study in 30,000 70-year-olds. So we won't get necessarily the data we want, but we will get some data, and then it will be up to health authorities and regulators to decide how that will be used. But then we can go into phase 4 studies, which does collect important safety data and effectiveness data. And from that, hopefully, we can look in reverse, so to speak, and decide how well the vaccines are doing. But obviously, it's important to start with the vaccine.
Right. We've got quite a few questions now, and I think we will endeavour to get to all of them. So the ones we don't get through before we have Paul speaking, we'll keep on hold to come back to. There was a question, someone was asking about the AstraZeneca Oxford vaccine, but Paul's going to touch on that.
Yeah, that's not what I talked about. No.
Yep. There's a question about the, if you could elaborate on the low immunogenicity of the RNA-based vaccines.
Yeah, well, see, traditionally when people have tried these in the lab and with animals, you get a very low immune response because you don't get much antigen produced. So that's one of the reasons Imperial College have looked at a self-amplifying RNA vaccine. People are looking at adjuvants as well. You will get an immune response. We just don't know if it will be enough. Now, so far, with some of these vaccines, the immune response has been surprisingly good. So, but most people, when they talk about immune response, talk about immunogenicity, which is not efficacy. So we have to wait for the phase 3 trials and the efficacy data. It's one thing to show immunogenicity in animals and humans, and it's another thing to show efficacy in humans with a phase 3 trial and a control group. We just have to wait and see.
Okay. There's a question around the fact that because of all these vaccines, we might be likely to receive more than one vaccine. We've had it in the past when we've had different sorts of Hib vaccines. What's going to happen? I mean, some people are saying, do we take the first one? Do we wait for ones that are more effective? Are they kind of compatible? How do we deal with that when, if you particularly need to have 2 vaccines, will you be getting the same vaccine, or is there another type of vaccine to give you that advantage? And we've seen that with things like the pneumococcal vaccine, for example, in older people.
Yeah, really, really good questions, and we just don't have time to answer them. It's with the warp speed idea and getting a vaccine out there as soon as possible, you haven't got the luxury to do those kinds of studies. If you had a 10-year kind of window, then of course you would. But with a 12-month window, there's, you're very limited in terms of your scope, funding and the rest as to what you can actually do. The aim is to produce a safe and efficacious vaccine that will hopefully prevent severe disease and hospitalizations. If we can achieve that with any vaccine, then that would be fantastic.
Okay, and I think I've got a chance for about 1 or 2 more questions because we need to get on to Paul, and we will try and come back to the other questions at the end of the session because we've actually left some time for that. So we have a question from one of my specialist colleagues, Catherine Mandel, who talks about vulnerable groups not mounting good immune responses. And so one of the endpoints is that can we reduce infectivity and transmissibility? And some of my GP colleagues have asked about that. If they get that, are they going to reduce their likelihood of passing it on to vulnerable people? So can you kind of elaborate on your comments about the transmissibility of the virus with these vaccines?
Yeah, and this whole problem of asymptomatic infection and silent infection and mild infections, where we haven't really got enough data of that fraction of the community that are carrying the virus. And we don't know, that won't be tested for really. The being able to stop transmission in the kinds of studies I've seen. It's a really important thing to be able to do, but as we all know, viruses cause subclinical infections. It's part of the reason they're so successful. I don't think prevention of transmission will be high on the agenda in terms of a clinical outcome. I think it'll be prevention of severe disease and hospitalizations. If it prevents transmission, I think we'll all be amazed. But I don't expect them to. Paul may have other views.
Okay, and perhaps one more question before we move to Paul. You made the comment, I mean, the Chinese have obviously got large numbers of doses, but it's a little bit disappointing that we actually don't have any of the data there. What's the likelihood that that will be provided? I understand the political climate's difficult, but often scientists are very collaborative, and it would be really important to know whether these vaccines are actually having an impact. Now, from an ethical point of view, if you're going to expose that many people to a vaccine, then we actually need to know there's benefit. Can you make any further comments? Some of it might be a little bit sensitive.
No, it is sensitive, and I don't want to be seen to be critical, but obviously it's a different sort of government to what we have. And transparency is not perhaps high on the agenda. And they'll use the vaccine, and whether they publish or not or share the data is another matter. But we don't really know what's going on in China. We only know what we're told. It's not going through the Lancet or New England Journal or Vaccine or something to be peer reviewed and looked at and understood. But of course, if there's data there, any data anywhere in the world will be incredibly useful to all the researchers and manufacturers around the world. So it is so important to share the data. That gives everybody confidence. But if they only use it in China and they want to export it to Australia, the community won't have much confidence in that unless all the data is made available.
That's great. Thanks for that. And look, I think we might need to move on to Paul now. I've still got some of the other questions there, so people hold your fire, and we'll get on to Paul's session. So I'd like to introduce Associate Professor Paul Griffin, who's the second speaker for tonight. And Paul's going to fill in some of the details that and complement the comments that Gary's very comprehensive talk has already provided. Paul is an infectious disease physician and microbiologist. He was appointed the Director of the Infectious Diseases at the Mater Hospital in Brisbane in 2013, and is the Principal Investigator at QFarm as an Honorary Research Fellow at the Mater Medical Research Institute and Queensland Institute of Medical Research. He's also an Academic Associate Professor at the University of Queensland, where he's currently involved in the trials of the Novavax COVID-19 vaccine with the Nucleus Network, and Paul's going to talk a little bit further about COVID vaccines before we have further questions. Can I hand it over to Paul, please?
Great, thank you, John, and thanks to the hosts for giving me the opportunity to talk, and thanks to Gary for great talks. It's a tough act to follow, but I'll do my best. You'll see in my outline in a moment that some of the topics I'm covering are a little bit similar to what was seen in Gary's talk, but I'll come at them from a slightly different perspective, so there shouldn't be too much actual overlap. I've a few relevant conflicts as well. So I do work for a company, Nucleus Network, which is a specialist phase 1 clinical trials company where my interest is vaccine trials. So I'm currently the principal investigator on 4 active COVID-19 vaccine trials and a few others in the works. I work for a few other companies that do some advising to do with vaccines, including Novatech, for example. And I also am a director of the Immunisation Coalition, who are some of the hosts this evening. So here is my outline. Looks like a lot to cover, but I should be able to do that well in the allocated time. So we heard a little bit about this already, that there's been a lot of people detracting from this vaccine development that we're doing in a hurry by saying there's never been a coronavirus vaccine. And that's indeed true. No licensed human coronavirus vaccines at the moment. We have had some success in animals, as we've already heard about. And I guess the thing that people often omit to discuss when they're talking about this is that we did have a lot of really promising candidates for SARS or SARS-CoV-1. And they crossed most platforms that we're going to talk about today, recombinant and inactivated, for example. A lot of animal models actually suggested, so that's small animals and non-human primates, suggested they might well achieve high levels of immunogenicity, but there were some complications there, including the immunopathology, which was thought to mostly be Th2 based, that we've already heard about, and the associated enhanced disease arising from that. And I guess the big difference with that first SARS issue we had was it was eradicated with infection control measures. And it was also a very different disease in that it was much more severe. And so we had obviously a much smaller magnitude there. So it's not that we didn't have any vaccines coming through for this, we got rid of it without requiring them, fortunately. Thought I'd quickly touch on clinical trials because this is something that's being discussed a lot, and a lot of people don't fully appreciate what those different phases of clinical trials actually look like or what the purpose of those is. So, obviously with preclinical studies, we want to make sure we have sufficient laboratory and animal data to move into clinical trials, particularly to show that whatever we're going to use in those clinical trials is not going to harm those participants, but is also likely to offer some benefit above and beyond what we have already. And obviously the extent of preclinical data we require to commence the human trials depends on a number of different factors, but we still need proper toxicology with the vaccine that we're going to use in the clinical trials, and also some basic immunology, at least to suggest it's likely to be effective. We often like to see animal challenge data before we move into human trials, typically where there's not the time pressure we have now, although that's been one of the areas it's been able to be relaxed a little bit in that the human challenge studies to show actual protection are often happening concurrently with these COVID vaccines. Phase 1 trials are fairly small, up to around 100 volunteers. And as we've already had some discussion of, they're usually a fairly narrow age range with very strict inclusion criteria, particularly given that the focus here is on safety, not really on efficacy. We often explore multiple doses, adjuvant combinations, but we do still collect some surrogates for immunogenicity to get a sense of whether they're working. But this is usually a secondary objective and not so much the focus. Phase 2, we get a little bit bigger, so often hundreds or 1 to 2,000 with the case of a lot of the COVID vaccines at the moment. Still a very strong focus on safety, but we start to move at least a little bit towards seeing how they work. And this is still predominantly laboratory surrogates of immunogenicity, which, as we've heard, doesn't correlate necessarily with efficacy or protection. But we also start to look a little bit at efficacy as well. And so I've chopped off a few letters there, but that's what that word is to the right. But they're obviously not powered sufficiently to prove protection, and that's what our phase 3 studies are. The inclusion and exclusion criteria are more liberal, and what we're seeing with a lot of the COVID vaccines at the moment, to address some of the issues we've already heard a bit about, is particularly the age criteria, but also some of the comorbidities start to be relaxed. We have older cohorts, for example, and some of the trials are even having a minimum numbers that we need to enroll in those elderly populations. So we should have some data at least that arises through that. We sometimes take a few doses or maybe different adjuvant combinations through to the phase 2, but a lot of that should have already been decided from the phase 1. And phase 3, which we're hearing so much about now, are quite large, and a lot of the studies are around that 30,000. Typically, they're international, and the focus there is on safety and immunogenicity, but obviously protection or efficacy becomes the key focus in that much more real-world environment. And the inclusion-exclusion criteria, again, to make it more real-world, become more liberal. And phase 4 is where we continue to monitor every dose of vaccine given to try and get as much information as possible once it's used more broadly. So here's a quick poll question from me. What are the typical primary objectives of the phase 3 SARS-CoV-2 vaccine studies? And I've got quite a few options here, so I'll run through them quickly. Need for mechanical ventilation, safety, the laboratory correlates of protection, tolerability, reactogenicity, preventing COVID-19, C and D, or B, D, E, and F. So I'd ask you to please cast your votes now. So there we go, a few votes for safety and those, a few others. And my questions are a bit easier than Gary's. So yeah, 75% got the right answer there, but it is still a bit of a minefield in terms of what those are. So I will come back to that in a moment. So here's a bit more on those objectives. So with the phase 3, the primary objective in most of the phase 3 studies is to prevent COVID-19. And this is where some of those discussions that we just had were alluding to. Often this is actually symptomatic PCR proven disease. So not necessarily representing everything we want to monitor in terms of the outcomes of these vaccines. There's still a strong focus in the phase 3 though on safety, tolerability, and reactogenicity. So that still remains a primary objective. Secondary objectives become very broad. So we look at laboratory correlates of protection. So those things we measure in the laboratory to see if the vaccine's doing things, not just antibodies that people often talk about, but also the cellular responses where we can. And we don't just look at symptomatic PCR-proven disease. Often in the secondary outcomes, we're looking at a whole lot of detailed clinical responses: asymptomatic infection, severe disease, emergency department visits, admissions, mortality, and a number of others. In terms of the phase 2, as I mentioned before, primary is typically the laboratory correlates of protection while still looking at safety. And in the phase 1, as I mentioned earlier, it's the safety, tolerability, and reactogenicity are the key focuses there. Just quickly touching on some of the differences with clinical trials for the COVID vaccines. One of the things is about how we're doing things so quickly. But the key point I want to point out there is we're not skipping any steps. A lot of people are worried we're not assessing these vaccines as thoroughly as we normally would, and I can assure everyone that's not the case. Without going into the details, there are lots of numbers on that, but one of the key drivers of the speed we're doing things at the moment is the funding, and there's been some great sources of funding, including CEPI that I listed there, the Coalition for Epidemic Preparedness, and they were already working on a lot of these projects before COVID actually hit. And then there's obviously a lot of generous government funding and now some advanced purchasing arrangements that are rolling out. We've certainly sped up our regulatory processes, again, not skipping any of the essential steps, but our ethics committees are doing things as close to real time as possible. The scaling of manufacturing, if you recall Gary's slide there, is often something that happens well down the process, but that's happening at the outset at the moment. So that's going to be something that should have been achieved by the time the clinical trials are done. Study design, we're trying to do adaptive designs as much as possible, which essentially combines the phase 1 and 2 in the one study, which can help speed up some of those regulatory approvals. And as I mentioned, with some expanding groups or elderly cohorts thrown in to include that important data. And with the preclinical data, we still want to make sure we're not potentially exposing any of our volunteers to any risk of harm. But the efficacy data, particularly the animal challenge studies, are often occurring in parallel to the phase 1 clinical trials. So we often get that. And certainly with the UQ study, the release of that excellent data did cause some confusion because people thought that should have been done before the phase 1, but it's definitely fine to do it that way around. Again, a very busy slide, and Gary sort of talked through a lot of this, but it was really just to highlight how long it's taken for some of the other vaccines that are around. And the table there is just to illustrate how quickly we've been able to get some of our studies initiated, from submitting to the HREC and going through all those steps in terms of review and independent expert reviews and approvals through to first participant, first dose. We've been able to do that in sort of 40 to 50 days, which usually that's many months, if not longer, to achieve some of those objectives. We have had a lot of challenges with these COVID-19 vaccine trials, and given that's what I've been doing, I thought I'd just quickly share some of those. We had a lot of trouble with recruitment, with a lot of people, I guess, scared of participating in these trials in the uncertain times, and with lockdowns and other things like that happening. Fortunately, we saw really high rates of expressions of interest with a lot of our trials, but then we had a lot of people basically not turn up. And when we dug a little bit deeper, a lot of that was fear relating to misinformation. A lot of people started to think they were going to contract COVID from participating in the vaccine trials. And there was a lot of misinformation about safety, essentially arising from some of those things I've mentioned in terms of skipping steps or not requiring the usual data to get into people. And so we had to educate people specifically on some of those things. And obviously we needed to make sure, particularly because we've conducted these trials, for example, in Victoria, as well as here in Queensland, where there was issues with the epidemiology at the time. So we had very robust protocols to keep our staff and our volunteers safe. One of the other challenges we've faced is trying to do a lot of the early phase trials in naive individuals. So we wanted to make sure they hadn't been exposed. And we didn't really have great assays for determining people were seronegative. So we had lots of false positives, which created a great deal of panic from a public health perspective. So we had to do a lot of convalescent testing and other testing to make sure we weren't finding all of these unknown positives in the community. And we also had to have a way of managing adverse events arising on those trials that didn't involve our usual process of bringing them straight into the clinical trial unit. Because if someone potentially had symptoms of COVID, we had to make sure we excluded that before we brought them in, to potentially not bring that into our unit. So we had to pivot a little bit there to be able to assess people remotely. One of the other things I wanted to quickly touch on in terms of clinical trials for COVID is the concept of challenge trials. Not something that's happening yet, but it is for COVID, that is, something well established for a lot of other infectious diseases, particularly malaria. At our unit, we've infected over 300 people with malaria to trial a whole lot of new interventions. And it's a great process in terms of hastening the development of those interventions. And it's happened for a lot of different pathogens. And you can essentially combine the phase 1 and adaptive protocol to actually go all the way through to getting some great efficacy data in a really controlled way. And it's not happening yet for COVID-19, but there's a really strong push. And there's actually a group One Day Sooner who are really driving this with a very strong international advocacy program. And the benefits are enormous of these sort of trials. We can look really closely at the infection dynamics. We have people in a really controlled situation without confounders. But obviously there are a lot of significant limitations here, including very complex regulatory issues and ethical issues, infection control issues, and public health, of course. You often do this on the really young and really fit people, so generalizability becomes a bit of an issue. And we typically like to have a rescue medication that we don't yet have really for this. And then all the COVID long issues, that the down the track problems people are talking about, do pose a risk to healthy volunteers as well. I'll just quickly flash up this tracker that we've already seen. Changes almost every day. And again, we'll focus on the sort of later phase trials, but so 12 in essentially phase 3 and 6, those limited approvals. And again, this is more of a framing slide, so very complicated, but you can see of the 4 platforms, there are many examples of all of them. And I'm going to touch on the 2 that Gary didn't cover in his great talk that just preceded mine. So the first one of those is the viral vectored vaccine. So this is where we get an unrelated harmless virus to essentially deliver a payload, which is a gene of one of the target antigens, into our cells. And so with a lot of these, we're using adenoviruses, human or simian, and they can be replicating, so they're able to reproduce or non-replicating, and they have some pros and cons. The pros are typically they produce high cellular and humoral antibody responses, and for a virus like this, it's what we need, sort of one of those balanced responses that do both. But they do have some limitations that pre-existing or even newly generated immunity against the vector may impact the efficacy. And while it wasn't talked about a lot, that is an issue with a lot of the popular viral vector vaccines, including the Oxford AstraZeneca one, and potentially some limitations in the scaling up. It's widely talked about that there's no examples of licensed vaccines using this technology, but in fact, the Merck Ebola vaccine is essentially licensed, and that's a recombinant replication-competent vesicular stomatitis virus vaccine expressing one of the glycoproteins from the Ebola virus. So that is one that's made it all the way through the clinical trial stages. And in terms of COVID-19 examples, obviously Oxford AstraZeneca and the J&J vaccine, as well as the highly publicized Sputnik 5 and CanSino. So CanSino is a Chinese adenoviral vector using that adenovirus 5. And as we already heard with some of the Chinese vaccines, a lot of those were approved for use very early on. And in fact, this one was approved for use by the Chinese military as far back as the 25th of June, even though the phase 3 is actually still going on. But this was one of the vaccines that made it into the media. I think it was a week or two ago now, where it was being released more broadly and people were queuing up to receive that. Although I haven't actually seen any published phase 3 results yet. And, we heard about some of the issues there. Sputnik 5 was the Russian vaccine using a similar adenovirus 5 and 26. Lots of talk in the mainstream media about this. Lots of memes arising, making some fun of this as well. And fortunately, we have to remember that we want to take our information in this context, not from press releases, but from reputable journals. And this was published, the phase 1 and 2 in the Lancet on September 4th. Still a fairly small study with 76 participants. And this, again, did result in a conditional registration certificate. This is the one that most people are talking about. And it's interesting that there are so many promising candidates and that this one tends to be such a focus. So the University of Oxford, a team there that have a great track record of developing or working on a lot of different vaccines. They've got, a chimpanzee adenoviral vectored vaccine called ChAdOx1. The phase 1/2 was published in the Lancet and looked very promising. I guess, again, one of the things that wasn't highly publicized from that was the reactogenicity element, including the fact that the placebo was already a relatively reactogenic licensed vaccine. And then in 2 of the 5 sites for that trial, they actually had to include a pre-medication to address some of those reactogenicity issues. So that might be something that impacts acceptance moving forward. Lots of subsequent studies, including the very large phase 3, which is underway. And this is the one that's talked about being very close to being able to produce some results from that, even as close as next month, potentially. Also, lots of pre-purchase agreements, including a prominent one in our country. And as I say, expected use is, emergency use expected very soon. Huge manufacturing capacity from a lot of good work in the scaling up process. And again, a lot of talk about the study hold for a likely unrelated issue, but it has resumed, and that wasn't deemed to be a significant safety concern to slow down the development significantly of that vaccine. Johnson & Johnson have a fairly similar vaccine program, although not as advanced as that one, using adenovirus 26. Great animal studies arising from this one. And one of the issues is about the background rates of neutralizing antibodies against the vector. And so with the adenovirus 26, it's thought to be quite low in Europe and the USA, but you'll see in some parts of the world can be quite high. And it's thought the efficacy of these vaccines, if you do have strong immunity to the vector, are likely to be significantly diminished. So whether this is a vaccine that has to be used, or any of these have to be used in certain populations or certain countries more than others, remains to be seen. Phase 3 is underway, and again, a widely reported pause. It wasn't a regulatory hold, and again, I think that's been alleviated now as well. Moving on to the protein-based vaccines. So this is an approach, again, that's been talked about quite a bit. And basically, we take a bit of the target antigen and make this from scratch in the laboratory. So I've heard people describe this as essentially having a 3D printer for proteins, and you can just basically take the sequence, plug it in, and generate your target antigen. But often if we do that in its crudest form, it's not all that effective. So a lot of the front-running candidates in this space have combined it with some kind of other additional technology to overcome some of the shortcomings of the crude approach. And they get called all sorts of things. Nanoparticles has nothing to do with microchips, which unfortunately some of the public have made that leap. But nanoparticles is one of the technologies, virus-like particles, and that's basically where you take a few of these antigens and assemble them into something that more closely resembles the virus without having any actual virus involved. And the highly publicized UQ's molecular clamp, which I won't go into that technology, but it's been widely publicized and a really clever feat of technology there. Pros, easy to manufacture. As I say, we can do that quickly from the sequence. No live virus involved, well-established platform. And because it's very specific in terms of what we put in there, the reactogenicity can be reduced, can be expensive, and often because of the relatively low immunogenicity, we do need adjuvants or multiple doses. Licensed vaccines in this space, human papillomavirus, hepatitis B, Haemophilus influenza type B, and many COVID-19 vaccine examples, and some are listed there that I'll just go into a little bit more detail of. The vast majority of these are actually being trialled in this country. So I've got a little section on vaccines in Australia where I'll cover those, but one of them that isn't so far is the Sanofi GSK one. And it's basically using the same technology they use in their flu vaccine. They have an established pandemic adjuvant technology, and they have a phase 1/2 that was relatively recently commenced. And as I say, all those other ones I'll touch on in a moment. I've got a lot of detail here under adjuvants, and I'll just go through this fairly quickly because a lot of people aren't completely familiar with what we're doing with these because it's a little bit more complex than the usual story. Obviously, the key role of an adjuvant is to boost the immune response, but with COVID-19, that becomes even more important, and there's some other functions thrown in. We want to vaccinate the whole world with this vaccine, and so an adjuvant not only boosts the response potentially, but means we can include less antigen in those vaccines, which means our ability to produce doses is increased, so it helps with the scalability. And as we've heard about a few times, the enhanced disease and immunopathology are likely to do with the type of response generated. And if we use an adjuvant that favors the other type of response, one that's hopefully effective, but less likely to do that, we can reduce those risks. And so that's certainly been a huge consideration in terms of adjuvant selection. How they work is difficult to sort of go through, and some of these mechanisms are not fully elucidated, but there's often multiple things that the adjuvants can help with, including keeping some of the antigen at the target site and helping to tip the immune system in the direction that we want it to go. Some of the adjuvants that I'll just quickly mention is, aluminium based, which is the most commonly used and in a few licensed products. One of the COVID-19 vaccines that include this, or two of them, in fact, the Serum Institute of India, as well as Sinovac. MF59, a proprietary adjuvant, which is an emulsion of squalene oil, is a potent stimulator of both Th1 and Th2, so the balanced response, which is thought to be less likely to induce ADE or immunopathology, and that's been used with the UQ vaccine. CPG, which is part of the Serum Institute of India vaccine program, and Novavax have a very well publicized proprietary adjuvant, this Matrix M, which again gives that balanced response, which should hopefully help to boost the response whilst reducing the risk of ADE. And with the protein or subunit vaccines, as well as inactivated, because they're relatively weakly immunogenic, we need the adjuvant. For the mRNA vaccines, we don't need that so much. And with the viral vectored vaccines like the Oxford one, the actual vector fulfills some of those same functions. But as I mentioned earlier, past exposure to that vector may be relevant. So a quick poll question: how many COVID-19 vaccines have been or currently are in clinical trials in this country? Answers are A, 3, B, 9, C, 5, D, 4, and E, 7. So a fairly big mix. The favourite was 3. A few people voted 9. Second was 5. A few people voted 4, and a couple voted for 7. So in fact, there are 5 vaccines that have been trialled in this country so far. The first one, and so these are in the protein or subunit group. So Novavax was the first vaccine to be trialled in the Southern Hemisphere that we gave to healthy volunteers at the end of May. So this is a recombinant spike protein that's assembled into this nanoparticle with that matrix M adjuvant. Results have been published in the New England Journal of Medicine and it looked very promising, with certainly with some antibody responses many-fold greater than we see with natural infection. We've expanded the age criteria. So we start with the phase 1 with a narrow age. We've added elderly groups. The phase 2 has commenced, many sites in Australia as well as internationally. And in fact, just a few weeks ago, the phase 3 started overseas, obviously, because we don't have any cases in this country really to justify adding sites, but that phase 3 is certainly well underway. The University of Queensland, I'm also the principal investigator on. Interesting product because it's a vaccine that's been developed in this country and then trialled in this country. And this again is a recombinant spike protein or S protein. And so as people know, that's how the main protein used for binding. So this is the one targeted by most of these using this approach with the so-called molecular clamp. And again, I'd encourage people to have a look. It's been widely publicized and a very clever piece of technology. First given to healthy volunteers on the 13th of July. And that's me there with a mask on talking to the first volunteer who's going to receive it. And this is an interesting project that was funded by CEPI that we actually started talking about probably 2 or 3 years ago, and CEPI awarded some funding a few years ago for us to plan to respond to disease X. And we'd planned a program of sort of practicing for this event, but it actually happened before we got to practice, and it's been great working with such a good group. And the preclinical data, as I said, caused some confusion because the animal challenge studies were publicized after the clinical trial was underway, but those animal challenge studies were very promising indeed. Clover is a S-trimer vaccine, a recombinant vaccine from a Chinese-based biotech, and that phase 1 has been undertaken in Perth. And people may have heard about a vaccine developed in South Australia at Flinders University by Nikolai Petrovsky, who is said to have completed a phase 1 with 30 actives that was said to have supported moving forward, but we haven't heard much about that one since some of those press releases. We have the Serum Institute of India, which we've just started, and we're about to start an oral vaccine as well from SimVivo. And the beauty of that one, in addition to it being oral, is it's temperature stable, room temperature stable, which will be great. So just some concluding slides now. So this is my prediction. Obviously, please don't hold me to any of this. And in short, there are no right answers here. What I expect to happen is we have a number of candidates looking promising in phase 3 that will have a moderately effective vaccine probably early in the new year, but it won't be perfect and we'll probably need boosting. Hopefully that'll be a longer duration than annually, but that remains to be seen. I think that'll be rolled out in a prioritized way. So perhaps healthcare workers, aged care, other first responders, maybe the military. And then in terms of how it's used in vulnerable patients, we've heard about some of these challenges already. Will depend on the efficacy and the information available in that population. Uptake will be a challenge to be addressed. When we have a vaccine that's initially released, we're not going to flick a switch and be the other side of COVID. We're still going to need some other intervention while we undertake that rollout and potentially generate more effective vaccines. We need to keep in mind that while we're here to talk about vaccines, it's not the whole story, and we need better tests as well as treatments as well. And hopefully we'll see iterative improvements in vaccines over time to overcome any shortcomings that may be there. I've got some quick answers to some of the questions that were sent in, and I'll try and do these in my allocated time. So a lot of people have talked about, is it true that vaccine trials won't tell us if they prevent mild disease or severe disease? And it's true that, as I said at the start, a lot of the primary objectives are about PCR-proven clinical disease, but in the secondary objectives or the exploratory objectives, we often look at a lot of other things, including asymptomatic disease, by doing regular virological assessments of participants. Is it true vaccines won't reduce transmission? And this does remain a bit controversial. While we're not focusing on transmission in the trials necessarily, obviously if we reduce case numbers, then the number of people infectious in the community will be reduced, so that will reduce spread. If we have any impact on symptoms, which is likely, that'll reduce shedding, so that will go some way to reducing transmission. And in the animal studies, a lot of them have actually shown reductions in viral shedding. So that will mean that hopefully they will reduce transmission. So again, we can't be confident because that data is not available in people yet, but it's likely it will help at least a little bit. This was a question raised before. If we have lots of generations of vaccines, how we know which will be compatible? The short answer there is that will work that will have to be undertaken as we progress through all of this, and I think will follow fairly quickly in time. Vulnerable populations, we are trying to include as many of those in the clinical trials as possible. Again, work that will follow behind doing it in the usual age groups and populations, but shouldn't be too far behind. Longevity and requirement for boosting, can't answer that yet. Most do currently use 2 doses. It's likely they'll be boosting. If it's too short, obviously that's going to potentially undermine the effectiveness of the vaccine program, but hopefully it'll be annually or longer. And someone asked if it'll be mandatory, and the short answer is probably not. I do think there may be some activity linked to being vaccinated in the fullness of time, but we'll just have to see. So just to wrap up, we are progressing quicker than ever before, which is really exciting to see, not skipping any of the steps that we usually do. All vaccine platforms have been applied. Some have been approved for limited or early use, but we have lots of phase 3s that should be coming through fairly soon. The scaling of manufacturing has meant that once the data is supported, we can start to use these vaccines in a prioritised way fairly quickly after, but there remains to be a lot of logistics that are going to be very complicated before we can roll it out to the entire population. So we're going to still need to have some fundamental mitigation strategies employed in parallel. And the last few points that I glossed over fairly quickly demonstrate that while we have progressed really well, there still are a lot of unanswered questions.
Thanks, Paul. That's great. We're sort of keeping pretty much the time, and there are still some questions here, so we'll turn to those. There's a comment about pediatric vaccines. We know that the spread in kids is much less, but obviously there are going to be kids with a range of health conditions and things there. Is there any, what's the status of pediatric COVID vaccine trials?
So something that's been discussed a lot, as far as I'm aware, there's not been a lot of trials so far happening in children. I mean, obviously, while there's a very strong push to try and have every vulnerable population or usually vulnerable population included in these trials, those imperatives are obviously balanced with the ethical implications of doing that. And I guess the thing with COVID that may have watered down the pediatric studies a little bit is we know that that's not a population that's as effective as, for example, the elderly, where there is a very strong focus. So, obviously the ideal thing with these trials would be to enroll, everyone, as many people as possible, but we do have to prioritize to get the information we need to keep progressing. And I think doing that in sort of the usual age ranges as well as the elderly makes sense, and that some of those pediatric studies will follow shortly after, hopefully.
So related to that, we have a question about, given that we have few cases of COVID circulating in Australia, how we know the vaccine's working, and will we be doing on that group? I think you partly answered that, because if we know it's working in other similar populations, but I'll leave that to you. And I guess the second part of that question is, will those who are getting this vaccine in Australia, will they be having regular serology to be monitoring measures of protection?
So yes, I mean, a good question. And that's why a lot of the sort of phase 2 and certainly the phase 3 studies aren't being done in this country because of our excellent control. We still have particularly the Novavax phase 2 that's happening here. So, we'll get more information from that. In terms of whether we know they're working in this population, well, that's why we have those surrogates of immunogenicity as well that enable some correlation where the epidemiology may not support getting sufficient cases to meet those objectives. So, so I think that'll give us enough information to cover as many populations as possible. I mean, similar to the age discussion, different ethnicities in different countries, different background rates of exposure to other things may be relevant as well. And so that's why the phase 3 studies are often international, involving as many countries as possible.
And as you say, doing the countries with high circulation so they can actually get enough endpoints. There are two questions probably on the Oxford vaccine, and they're in some way related. One is a comment that it actually, why is it getting a whole lot of attention? And the other question was asked about what they regard as the indifferent primate study results for the Oxford trial. Could you comment on those two, please?
Yes, thanks. Yeah, all great questions coming through. I don't really know why the Oxford vaccine is getting so much attention. I mean, I think a lot of it comes from it being, temporally quite close to the front. So it was one of the first ones, but, Moderna certainly were in clinical trials before they were. So I think it's a combination of things. I think it's the country from which it's arisen. I think it's the speed at which it's progressed. I think it's the promising results, and certainly the phase 1/2 study was very promising. But in that same edition of the Lancet, there was a Chinese vaccine study published that was very similar with promising results as well. And the media response was a little bit disparate in terms of talking up one and not talking about the other one so much. So we know with COVID that that the media have had a big part to play in what's been released, and it doesn't always make scientific sense in terms of what the media are focused on. But it is, don't get me wrong, a very promising vaccine, and we should have some phase 3 data fairly soon. But as alluded to in the question, it's not the only vaccine we should be looking at. The other question about the indifferent animal studies. I think there were some studies in animals that suggested maybe the enthusiasm was a bit exaggerated, but I think in the end, we're going to see varying results from different trials. And what we really just need to do is make sure that it's safe and effective enough to progress through the clinical trial stages. And when we've got phase 3 data, we'll make that assessment with that data in hand. And, it's very likely still that one of those 12 vaccines in phase 3 trials won't meet the primary objectives and move forward to use beyond that. And maybe a significant proportion of them, that might be the case. But as I say, I think we need to obviously eagerly await those results and respond accordingly when we've got the data to make an accurate assessment.
Using Chairman's privilege, I'd like to ask a question that a couple of my colleagues have raised, is that GPs are going to be very important in implementation of this, and I suspect we'll have another whole webinar on that. We know with a lot of vaccines in the population that if a GP recommends it, they'll get it. We already have 2 letters to the editor in the Lancet, one back in April and one in June, and suggesting during lockdown, it looked like about 85% of people would get the vaccine, but as soon as things opened up, in a separate group asking parents, the uptake dropped to 75%. So my kind of question is, the reproductive number determines the number of people to give us herd immunity. The kind of figures being bandied around as 60%. What do you realistically think we have to get coverage up to? And again, if we want to look at a parallel experience, look at the app. We've had 7 million people use it, and unfortunately, it hasn't delivered all the things we've wanted to. So let's have a look at the numbers. What's a realistic target that will give the protection as one strategy to kind of get this disease under control?
Yeah, that's a tricky question, and a lot of people have bandied around numbers, and I think a number probably doesn't tell enough of the story. I mean, it really depends on what we're expecting this vaccine to do, and obviously it's going to depend on how effective the vaccine is, how long lasting the protection is. It's going to, in some ways, be dependent on the epidemiology at the time. So it's really hard to answer. I mean, obviously we'd all like an ideal vaccine that's 100% protective in 100% of the population with a single dose. And, what's clear is we're not going to have that. But even a vaccine that's modestly effective in parallel with some ongoing infection control measures to reduce transmission will help. So I don't want people to think that if it's not 70% effective, it's going to have zero impact on all of this. I think, a vaccine that's 50% or greater, used appropriately with concurrent infection control strategies still employed, will be helpful. And I think the thing is, we're not going to stop with this first round of vaccines, unless, of course, one is perfect. We're going to have iterative improvements throughout. We're going to learn a lot from how successful these first ones are and hopefully improve on those so we get closer to a perfect vaccine. So, I think we shouldn't be disappointed if it's not 80 or 90%, as some people are suggesting. I think any vaccine that gets through a phase 3 with perhaps even 50 or 60% efficacy will still be a big step in the right direction.
Thanks, Rob. So just related to that is about the general population and giving them some information about safety, because clearly that was an issue in the people sampled here. Do you want to make any other comments about how we let people know about that? Or, I mean, that may end up being a topic we tackle in another webinar when we actually know which particular vaccine we're going to be offering people and can be a bit more focused in on the specific, the efficacy of that, or sorry, safety issues of that particular vaccine.
Look, again, that's one of those things that, you know, the phase 3 data in large numbers will be really important in terms of the safety as well. And, you know, the ones that have made it to phase 3 have certainly had phase 1 and 2 data to support progressing into phase 3. And certainly, you know, there's no ethics committee that's going to permit a phase 3 to commence without phase 1 and 2 data suggesting it's both safe and effective. And then in terms of exactly how safe it is, I think we need to wait till we have the phase 3 data to be able to make very informed commentary around that to see exactly how safe it is.
I think we're getting pretty close to time, so I'm just going to just take on board 2 more questions. One question was, if the studies are fairly short-lasting, now how can we be sure that the volunteer is actually exposed to the virus? So I think that's a sample size issue to be confident that we can get an estimate.
That's right. And, you know, in the countries that have very poor control of this at the moment, having 30,000 people in those studies, a significant proportion of those in a fairly short space of time will be exposed. So that's about choosing the right sites for those phase 3 studies.
Okay. And then the final question, some viral vectored vaccines are non-replicating. Is this also true in immunocompromised people?
Are we using the non-replicating ones in immunocompromised? Sorry, is that the question? Hang on.
It was actually more about whether there's an issue in immunocompromised people. I don't know whether that was in the, we need to clarify that with the person asking the question, whether it was actually in that target group, because I don't think they're going to be a large number of people.
Yeah, I mean, basically not. It's even though the vector's replicating, it's not like a lie, like you know, how we often worry about the MMR, for example, in immunocompromised people. Sorry. So no, I mean, and again, it will depend on the data that's generated from those trials, but it won't be something, hopefully, that can't be given to immunocompromised people. But, you know, it's one of those things that once we get past the phase 3 to suggest using it more broadly, that hopefully we'll do some studies on immunocompromised people to understand more exactly how those vaccines look in that population as well.
Thanks. Gary, is there any other of those questions? Amanda, both of you have done a terrific job in answering them, and I have to apologise to those we haven't got to, but as we've already flagged, we're very happy to take them on notice, and we are looking to have a Q&A sheet, and we appreciate that some of the answers are going to be coming in. So while we can't give them an answer now, we'll try and give you a timeline. Gary, before I kind of wrap up, and thank you guys, is there anything you'd like to further say, or any other questions you'd like to comment on?
No, not really. I think the one on immunocompromised is a good one, but as I understand it, and Paul can confirm, the adenoviruses are actually engineered so they cannot replicate. Okay, well, look, I'd like to thank both speakers. It's been an encyclopedic talk tonight. I think we've all had a lesson in immunology, and I think what's quite clear is that there'll be a need to do this again. It's a work in progress. It's been a very lively session. The 2 presenters have done a terrific job, covered a lot of ground, and I think have done a good job of answering all the questions. Thank you, everybody, for participating and for your questions. Please don't forget that you'll need to fill out your feedback sheets. I'd like to thank the Immunisation Coalition for running this, and our partner in organising this webinar, Prax Hub. So as I mentioned at the start, the webinar will be available on the Immunisation Coalition website in a couple of weeks' time, and the Q&A question and answer sheet will also be available. So after we've wrapped up, you will actually get a questionnaire online to fill out for your CPD points, please. Please do that. And many thanks for participating, and good night to everybody.